So we have 5 great speakers. The first one is Paulina, and she's going to talk about the enhanced survey. Thank you.
Hi everyone. So my name is Paulina, and I'm here to talk about an ongoing program of work that we've named the Enhanced Project. We've got a number of collaborators. We've been really interested in medical specialty college set requirements for research and how these might be driving research waste. Can I just get a quick show of hands? Who's in health and medical research in the room? Who's been approached by a clinician at some point to help them to do research for their medical training? Okay, so you guys might understand where I'm coming from. So we published a curriculum mapping review a couple of years ago now, and we were basically looking at what the research requirements as they were stated by the colleges. And we found that there was a really large emphasis on leading research and not so much of an emphasis on learning how to do research and being supervised by skilled researchers. So we then wondered, do these mandatory requirements incentivise research waste? And we were also concerned about the potential, educational experiences of the trainees. So we did a survey across Australia and New Zealand for current and recent past medical trainees, and some of these went on to do in-depth interviews about their experiences. I'm just going to do a very small slice of our results, which is about it leading to research waste. So what did we find? We had most of the trainees said that they did a project of some sort, some did only one. The vast majority only had to do one project, and some had to do more. So when we asked them about the conduct of each individual project, we had 67 individuals tell us about 92 projects. So that's what this data is about. When we look at how they developed their questions, 74% had a look at the literature or systematic review prior. 41 generated their question on their own, so that's not in clinical discussion, and very few were part of ongoing projects. And only 8% involved consumers in their research, and none of those consumers were involved in the generation of the research question. When we asked trainees how they developed the study design, the vast majority developed it completely on their own with minimal to no help from others, half the trainees. And 75% developed a protocol a priori, that's what they told us, but 70% of those protocols are not publicly available. When we have a look at when trainees did their projects, you can see the green bar there is doing their project in their own time. So the vast majority of them are doing their projects outside of work hours and not during protected time. And that's across all the colleges except for the Royal College of GP. And they have a special program that has some funded research time, and they're partnered up with the university supervisor. And 50% of the projects remained unpublished at the time of the survey. We also asked the trainees to upload a copy of their manuscript. When we were consulting with the colleges, they don't keep the projects that they require them to do in some of the colleges, and others do, but we need to get individual consent, so this was the best way that we could go about it. And we had 34 participants upload their manuscripts. And we did 2 rounds of data extraction. The first was to sort of work out what we're dealing with, and the second round was to do quality assessment on study design and reporting. So we felt that of the 34 studies, about 70% had adequately considered the literature in their introduction, 90% provided a sound study rationale, and 82% provided a clear question. That means that just under 20% didn't. Yeah, so the top study designs are there. We found it really interesting that 40% of the studies didn't even mention the design approach. That was quite telling in the reporting. 15% of the studies that needed to do a sample size calculation actually did one, and 25 of the 28 studies were deemed to have low to moderate quality. This is a slightly nicer view of the rest of the population potentially, because most of these are published. All were published in peer review, and one of them was potentially predatory. So what happened with the quality of reporting? So you can see here, we've put together the 27 of 28 studies that we did for quality of reporting. That's because they could map across these. One was a clinical guideline, it mapped across different areas. We can see in introduction and discussion that the clinicians did really well, because it's part of their clinical knowledge, but not so well in areas that require research knowledge. And this is reflected then in the title and abstract. So, do mandatory projects lead to research waste? Probably. And I'd like to thank the whole team, our funders and the participants, especially those that uploaded the results.
So our next speaker is David, and he's going to talk about software citation taxonomy. Thanks very much.
Yes, I'll just give you a second, like 4 minutes. It's one of these working slides.
So I'm David, I'm a research fellow at the University of Melbourne. I've been a thorn in Fiona's side for the past few years. So I'm here today to talk about a side project I'm working on, which is a software citation taxonomy, or SofCiT. So it's a protocol to improve research recognition and transference. So I'm working with this great team of folk, most of the faces you've seen around the room, and to put it up front, most, if not all of us, are people who would benefit from this sort of protocol being used. Technology moves rapidly. Academia doesn't. You just seen in that last mini note that after a decade or so of work, we still haven't solved the problem with data sharing. Academia is lagging behind in how it handles software. Technology change is giving us new ways of collecting data. So ecologists, we have environmental DNA sampling, using drones with heat-sensitive cameras. We've got bigger computers, we can analyze bigger and more complicated data sets. But how academia recognizes software is still lagging behind. So where this impacts people the most is early career researchers, graduate students, professional staff, so like embedded research, academics. We're the ones doing the software development, maintenance, but it's not being recognized as an equivalent primary outputs like you would for a traditional research paper. There's been some movement towards that in recent years with stuff like software papers. But when you're doing just traditional paper outputs, once you're done, you get your paper, you move on to something else. If you're still working on the same sort of thing, it becomes a second paper, you're getting another output. Software development doesn't end the moment you get your initial release. You've got years of maintenance, support, adding new features. So while we might get new versions of software, those aren't being recognized as traditional outputs. Software development has kind of had to mold itself to suit academia as opposed to academia changing to suit the fact that software is now such a big part of it. So while we're getting these software papers, because we're still academics and everything needs to be novel as a condition of getting a publication, new versions of software generally aren't meeting that criteria unless you're doing some major development to something, like Shinichi with the Orchard package, got the 2.0 paper, or something like the Unmarked package, which is kind of like a ubiquitous name within its sort of modeling approach. And like that's got a 10-year retrospective of like all development in the field, whether it had to do with that software or not. But it also impacts transparency. So this is a paper worked on with PhD students at the University of Melbourne, and they were looking at doing computational reproductions of papers that have put their code and their data out. So on principle, you would expect to be able to get the same results, but when quite often we're not getting either the same results or we can't run the code. The same as we saw it in Tom's talk, and that's because the way software is reported in papers isn't adequate. If you cite a paper, someone can go off and find it. If all you do is have in the method section sentence that says, we used such and such package, you're missing important details like software versions, the environment to run, and they all have big impacts on whether you can reproduce results or not. So that's where SofCiT comes in. For this, take inspiration from a few different places. So Force 11, we're using their software citations guidelines, and then the way we're presenting it is kind of a mashup of like the contributor roles taxonomy and the methods reporting issues of transparency. So what we're proposing is that all research software that you're using or calling directly that impacts your research needs to be cited. So you decide words, things like that. But like language you use, packages you call. Software citations need version numbers, otherwise you're missing. It's just like, here's the paper I use, we're not going to give you the title or any of the authors that did it. Any software that's being called indirectly, like dependencies, you wouldn't be called in your main paper, but you still report it, and there's a variety of ways you can approach this, but I'll have to skip those for now. The key ones are any software that's critical to reproducing your results need to be explicitly stated in the text of your method section. No one cares which package you use to make your figure, or which regression framework you use to come up with your numbers is important. Finally, all software is including like machine readable statements, much like a credit statement for author contributions in the paper. And then what this might look like is in text. It explicitly goes in a sentence. Software statement is this machine readable thing. I'll skip that one for time. And that's it.
Thank you, David. Our next speaker is Danielle, and she will be talking to us about non-verifiable cell lines in cancer research papers. Thank you, Danielle.
Hi, my name is Danielle. I'm a part-time research assistant and undergraduate student majoring in animal science at the University of Sydney. I'll be sharing our work on non-verifiable cell lines in cancer research papers. This is the first time presenting this work as it's still ongoing. Cell lines are used in biomedical research to study the effects of treatment or gene involvement in cellular functions and disease. They're identified by a series of letters and numbers, but the nomenclature is not intuitive and often confusing, and it may not reflect cell line biology. Additionally, cell lines have often been known to be wrongly identified due to contamination or misclassification. This can lead to incorrect interpretation of results and can mislead translational research. So we manually verified reagent identities from a cohort of original articles published between 2020 and 2022 that mentioned human, cancer, and MIR145, which is a short non-coding gene often studied for its potential involvement in cancer. We also verified published cell line identifiers using Cellosaurus, which is an online cell line encyclopedia, to check that they match their claimed identity. Around a third of the articles contain misclassified or contaminated cell lines, and we also found 5 non-verifiable cell lines that are not recognised human cell lines in Cellosaurus. To assess the prevalence of these non-verifiable identifiers, we conducted Google Scholars using identifiers as search terms. Some were similarly named to known human cell lines or known misspellings, and so we also conducted searches carrying these with the non-verifiable identifier. We later included short term repeat and STR to identify publications claiming authentication of these cells. Through this process, we screened hundreds of articles and found a further 8 non-verifiable identifiers. These articles fit into 3 main categories of varying significance. Firstly, many may be typographical errors. For example, the non-verifiable identifier was used interchangeably with a similarly named cell line. Concerningly, many similarly named cell lines are known to be contaminated, and so at best, these papers will describe wrongly identified cell lines. Secondly, there were articles that only referred to the non-verifiable identifier, no similarly named cell line. And finally, articles were found mentioning the non-verifiable identifier and a similarly named cell line, but the 2 were described as independent cell lines. We found over 200 articles referring to 6 non-verifiable identifiers as independent cell lines. At this stage, we've identified 13 non-verifiable identifiers, and we've finished screening articles for 8 of these. We've been unable to verify their origin nor find establishment descriptions, and they do not appear to be in any claimed cell line repositories with accessible catalogues. Some articles claim to have generated short tandem repeats for these identifiers, yet none have been identified. Although some non-verifiable identifiers may represent misspellings, it seems a subset of these have assumed new identities as independent cell lines. Regardless of the error type, these mistakes render the described research unclear and pose challenges to research reproducibility. Further investigation is necessary to clarify their identity and to identify others. Urgent adjustments are required for cell line reporting and published descriptions, and we recommend flagging manuscripts with non-verifiable identifiers for further investigation. This should create a zero-tolerance approach towards identifying misspellings, as the article should be retracted if authors cannot provide authentication or establishment data. We also recommend manuscripts use at least 2 identifiers when reporting cell lines, such as the research resource identifier, to improve transparency and clarity of the research conducted. I'd like to acknowledge my colleagues for their contribution to this work and NHMRC for their funding support.
Thank you, Danielle. And so our next speaker, I think we have Kabir, who is with us, and he's going to be talking to us about AI in interdisciplinary research, which remains advancing in science. Thank you.
Hi everyone, I'm going to start by quoting 2 people from computer science. The first one is Kabir. He says, if you use to achieve our purpose, a mechanical agency with whose operations we cannot interfere once you set it going, then we better be sure that the purpose we put into the machine is the thing that we truly desire. And recently, Brian Christian extended this by saying this has become one of the central concerns for the field of AI. We now know it as the AI problem. Welcome to this talk on AI in interdisciplinary research, bridging domains and advancing open science. My name is Kabir. I'm a research lab specialist at the University of Byrne in Open Data Management Platform. We're a team who do data-intensive research across several domains in the university, and my work focuses on AI. Right, so I'm going to start with a couple of quick questions here. Raise of hands to those who used ChatGPT over the last year. Oh, that's great. And those who use ChatGPT for research and have kind of like a positive sentiment or positive sentiment in general. Much fewer, less maths. What I want to start by saying is that AI may still not align with expert opinions, and it's like there might still be some flaws. But what's clear to see is that in the last decade or so, we've seen massive improvements in this area. With AI maturing, we can see that it's kind of integrating in several domains, causing it to change traditional research methods. Basically, with AI, we can analyze heaps of data sets, and then kind of like the sheer volumes of which humans cannot, and then generate insights which, when combined with domain knowledge and domain expertise, can result in significant outcomes in the domain. The other aspect to this is that AI models are now getting tested against real-world problems a lot more than before. And this kind of like not just challenges AI, but kind of brings forward several flaws in the models that's not seen in traditional benchmark approaches. And this is good because this is kind of helping us to refine AI research in general, and then also hopefully in the future gaining more trust from these domains. Finally, when we're doing interdisciplinary research using AI, what happens is that we need a way to communicate our findings and outcomes. So we kind of this kind of leads to the flourishing of open science practices with data visualization, websites, interactive platforms, open source code, and open source data that we've discussed a lot in this conference already coming out of it. So finally, I'm going to talk about a couple of case studies that I've worked on that kind of highlights the stuff that I've talked about. The first one is analyzing heaps of speeches given by Malcolm Fraser, a significant figure in Australian politics. And the goal of the collaboration was to kind of understand his change in rhetoric over time. To do this, we ended up creating a website where we visualized several of the outcomes of the model and then combine that with interpretation from our subject matter experts. Along with that, we kind of recently published this paper in a conference called Natural Language for AI, where our findings suggest that if we had just used GPT-4 and did not have subject matter experts, then we wouldn't reach the same conclusion as we did using it compared to using experts. The thing is, GPT-4 kind of missed out on certain few teams, which kind of could prove to be significant at understanding Fraser's rhetoric. Finally, the other collaboration that we are working on right now is predicting the land cover change in savanna in Northern Australia. To do this, you kind of build an AI algorithm that kind of like looks into the land cover data for the last 3 decades in northern Savannah. Also, you've got like heaps of features that's selected by experts. We kind of managed to get really good accuracy, around 80% at the moment, and we intend to use this for climate smart policy for that particular region. We're committed to like transparency, which is why we'll open source the code and the data that we use to model. We hope that that helps create transparency, but also extend or analyze the work that we have done. Thanks, that's it for me. If you have any questions on what I just said or want to discuss collaboration, that's my email ID, and please reach out to me.
Very well for time. So our last speaker is David Smailes. He's joining us online, and he'll be giving a citation analysis of a flawed meta-analysis for that studied the efficacy of cognitive behavioural therapy for bipolar disorder. David, are you ready to go?
Yeah, can you hear me okay, folks?
We can hear you. Might just turn up.
Perfect. Right on. So yeah, sorry for the long title. So this is the meta-analysis that we're a bit worried about. It's by Chang et al. It got published in 2017 in PLOS One. As the title explains, it looks at the efficacy of cognitive behavior therapy for bipolar disorder, which is a pretty severe mental health problem. What you might be able to see is that in this meta-analysis, which looks at the effect of CBT on depression in bipolar disorder, you can see that there's a medium-sized effect and it's statistically significant, but hopefully you can also notice that there's an enormous outlier in this forest plot. So this effect size here, from Gonzalez Isasi et al. Is minus 5.015, whereas the next largest effect size is minus 1.641. And then similarly in their meta-analysis of how well CBT reduces mania severity in bipolar disorder, we see almost exactly the same thing. A medium-sized effect overall, minus 0.581, and that's statistically significant. But again, this pretty obvious outlier, again extracted from Gonzalez Isasi et al. In 2010, and the effect size is minus 7.862, which is enormous in comparison to the closest effect size, which is up here, minus 1.448. So in the paper, in the supplementary materials, Chang et al. Show that if you exclude this effect size from these two meta-analyses, the overall effect of CBT on bipolar disorder becomes non-significant, but that is kind of downplayed in the text of the manuscript. Now, the Chang et al. Studies being cited reasonably frequently, about 129 times now. And so over the summer, we thought, well, let's have a look at the nature of those citations. Are they kind of appropriate, sensible, critical, caveated comments where people go, maybe there's some evidence that CBT works for bipolar disorder, but there are problems with one of the meta-analyses that suggested this? Or are they really uncritical citations where people go, CBT works for bipolar disorder, see Chang et al. And we were especially interested to see if most of the citations are the latter, because if they are, then that's more evidence that science isn't self-correcting. So a few talks ago, Tom Hardwick mentioned that this is meant to be something that science does, but in psychology at least, we've got lots of evidence that psychological science doesn't really do that. If you look at things like citation patterns for original studies, which report some kind of high profile effect, and then the replication failures that kind of have followed some of those effects, we don't really seem to be self-correcting ourselves in psychological science. So we developed a pretty simple coding scheme. There's one or two extra parts. This is just part A, which is kind of the most important bit. We scored a paper as 1, where the authors used Chiang et al. In kind of an uncritical way to go, CBT works for bipolar disorder. We scored a paper 2, where the authors noted some problems with Chiang et al. Or kind of gave a critical or caveated citation. And we scored a paper as 99, where Chiang all was cited in some way which wasn't really relevant for our purposes. So, and there was kind of a neutral citation. Maybe folks just say, oh, the number of meta-analyses about bipolar disorder have increased over time. And what we found were 18 instances where a paper was scored 99, where the citation was sort of irrelevant to kind of what we were particularly interested in. We found only one instance where a paper was scored 2. So this is where there's that kind of really critical caveated comment about what we can conclude from Chiang et al. And the remaining 100 or so papers were scored as 1. So where there's been this uncritical, kind of uncareful citation of Chiang et al. And so we think this is another example of psychological science failing to self-correct, a little bit different to some of the examples that have been published before, because it doesn't rely on people looking at like a replication failure. It just kind of is there smacking you in the face when you look at those forest plots. And we think we might worry about this a bit more than normal because of the clinical implications of this study. The one caveat that I should add really is that this is the first run of this coding scheme, the kind of data I've just presented today, and we're testing the rate of reliability of the coding scheme at the moment. Okay, that's me done. Thank you very much, folks.